In this video, we will talk about the gut microbiota, and in particular the role that the gut microbiota plays in multiple sclerosis. People with multiple sclerosis often show gut dysbiosis, that is, the bacteria that make up the gut microbiota are not balanced or are distributed in a different way than they are in healthy people. And this dysbiosis could contribute to the development of multiple sclerosis through different mechanisms such as activation of immune cells, disruption of the intestinal barrier, induction of molecular mimicry, or activation of humoral immunity. Some bacteria from the gut microbiota favor the activation and differentiation of pro-inflammatory lymphocytes, thus contributing to the development of inflammation, while other bacteria favor the activation and differentiation of regulatory lymphocytes and therefore contribute to the control of inflammation. A dysbiosis consisting of an increase in bacteria capable of inducing inflammation and a reduction in bacteria capable of controlling inflammation could contribute to the development of multiple sclerosis. A healthy and diverse gut microbiota helps maintain the integrity of the intestinal barrier, while an altered gut microbiota, like the one often found in people with multiple sclerosis, can disrupt the gut barrier. The disruption of the gut barrier can allow the translocation of bacteria into our organism, where they can activate immune cells and induce inflammation, and produce substances such as lipopolysaccharides, LPS, mediators, or toxins that can access organs far from the intestine, such as the brain, through the blood, where they can disrupt the blood-brain barrier and therefore facilitate the entry of immune cells such as lymphocytes into the central nervous system, directly influence the maturation and function of cells present in the brain, such as microglia and astrocytes, and even directly damage the brain. One of the hypotheses for the onset of multiple sclerosis is that T cells, which are cells of the immune system, recognize through their receptors bits of proteins presented to them by antigen-presenting cells in the central nervous system. After this recognition, T cells are activated and produce mediators such as cytokines that attract other cells and components of the immune system, inducing inflammation. An inflammation that ends up damaging the myelin that covers the axons of neurons and thus inducing demyelination. It is important to note that in order to be able to enter the central nervous system, T cells first have to cross the blood-brain barrier, and only memory T cells can do so, that is, T cells that have previously been activated outside the central nervous system in the periphery. T cells are generated as non-activated cells, which are called naive, and in order to be activated, they need to recognize with their receptor bits of proteins for which they are specific. These naive T cells can be activated in the periphery because they recognize the same protein that they recognize in the central nervous system, for example, because the protein has been taken up by antigen-presenting cells, or because they recognize something very similar from, for example, pathogens such as viruses or bacteria that infect us, which is known as molecular mimicry. After this recognition, naive T cells are activated, proliferate, become memory T cells, and by mechanisms that are still unknown, cross the blood-brain barrier and enter the central nervous system. A few years ago, our group discovered that in some people affected by MS, the T cells which are present in the brain recognize bits of an enzyme called GDPL fucose synthase, which allows the synthesis of fucose, a very important sugar for the brain. Curiously, these T cells, in addition to the human enzyme, also recognize the enzyme from some bacteria of the gut microbiota, and in particular from Akkermansia, which, if you remember, is the bacterium that has been found to be increased the most in people affected by multiple sclerosis. This finding led us to hypothesize that Akkermansia could activate naive autoreactive T cells in the gut by molecular mimicry. In the gut mucus, there are different types of bacteria. Bacteria that can only feed on fiber in green. Bacteria that, in addition to fiber, can feed on intestinal mucus in orange. And also pathogenic bacteria in purple. After a diet very low in fiber and for a long period of time, the bacteria that can feed on the mucus expand while the thickness of the mucus is reduced, which favors pathogenic bacteria to gain access to the gut epithelium and damage it, inducing inflammation. Interestingly, Akkermansia is one of the most effective bacteria at feeding on mucus when there is no fiber in the diet. A very low fiber intake may therefore increase the amount of Akkermansia and induce inflammation, which may facilitate the activation of pathogenic autoreactive T cells in the gut. In addition, it has been seen that a disruption of the gut barrier is associated with a disruption of the blood-brain barrier that can facilitate the entry of pathogenic T lymphocytes into the central nervous system. Antibodies are also key molecules of the immune system in the defense against pathogens. Antibodies cover viruses, bacteria, or infected cells, facilitating their recognition by phagocytes, which are cells capable of phagocytosing them, that is, of eating and destroying them. In addition, antibodies facilitate the binding of another component of the immune system called complement, which, when activated, creates holes in the membranes of pathogens and infected cells while facilitating their recognition by phagocytes. There are different types of antibodies. IgM antibodies that are the first to be produced in response to an antigen, IgG antibodies that are the most abundant, IgA antibodies that are present in mucous membranes, such as the one in the intestinal tract, IgE antibodies that are involved in allergy and defense against parasites, and finally the IgD antibodies whose function is still quite unknown. All these antibodies are present in blood except the IgA, which is found on mucous membranes, including those of the gastrointestinal tract. The production of antibodies begins in the lymph nodes, which are formed by follicles and germinal centers. In the follicles, B cells become activated and, with the help of T cells, differentiate into short-lived plasma cells that produce IgM antibodies. Activated B cells can migrate to another area of the follicles called the germinal center, where, after interacting again with T cells and receiving their help, they differentiate into long-lived plasma cells that produce more advanced antibodies that can be IgG or Ig. In the intestinal lymph nodes, known as Peyer's patches, these plasma cells produce IgA antibodies. IgA antibodies are very important for the control of the gut microbiota. When the gut barrier is disrupted as a consequence of a dysbiosis and translocation of bacteria into the body occurs, IgG antibodies are also produced to control these invading bacteria. IgA antibodies specific for gut microbiota bacteria, such as those specific for Akkermansia, might play a role in the development of multiple sclerosis. Since B cells that produce these antibodies can migrate from the gut to the central nervous system and are found in the brain of people affected by multiple sclerosis, Akkermansia-specific IgG antibodies could also play a role in multiple sclerosis, since they are more abundant in the cerebrospinal fluid of people affected by multiple sclerosis, and their concentration correlates with the Disability Index Expanded Disability Status Scale. With all the information that we explained in this chapter, we hope that we could convince you that the gut microbiota plays a role in multiple sclerosis.